Hello everyone, my name is Tyler Tierney and I am the AmeriCorps Watershed Ambassador serving in Watershed Management Area 5. Today, we are at Brass Castle Creek in Washington Township performing a stream assessment. I want to introduce you all to the fun procedure we follow when carrying out these assessments and talk a little about why we are here performing them. The New Jersey Department of Environmental Protection assigns us GPS coordinates that lead us directly to the streams where they need us to collect data. We enter the stream seeking out benthic macroinvertebrates to use as indicator species on the stream's health. The term benthic means below the water surface. Macro means we can see them with our eyes, and invertebrate meaning without a spine. Each of these tiny bugs has a different tolerance level to pollution, so our goal is to collect and record a healthy, biodiverse sample, which would indicate that the stream is in good health. We want to collect and record macros from each of these three groups shown in this image. Green indicates pollution intolerant, meaning little chance of survival if the water quality is low. The next group is pollution sensitive. This means they can survive in a stream slightly impacted by pollution. Lastly, we have pollution-tolerant macros. These macros can survive in a stream even if the water quality is extremely low. So, if we only find pollution-tolerant macros such as leeches, aquatic worms, or midges, the stream will be listed as stressed and in need of further research by other professionals. The NJDEP tasks us to complete both a biological and habitat assessment in our streams. The biological component involves collecting the macroinvertebrates from the various microhabitats found within the stream. The habitat, or visual assessment, requires us to score the health of the riparian zone, which is the environment directly surrounding the stream, along with the characteristics below the water's surface as well. Now I want to share with you some of the equipment and how we use it during our stream assessments. Our main tools are waders, which keep us dry and safe from the water, and a D-frame net. The D-frame net has a long handle attached to a metal frame in the shape of a D. Attached to that frame is a seine net that allows water to pass through it but captures the macroinvertebrates that we are seeking out. While facing upstream, we place this net directly in front of our desired sample zone, scrub the rocks with our hands, gently kick our feet around, disturb the habitat, and allow the macros to flow into the net using the current. We generally want to seek out areas called riffles to find most of our macroinvertebrates. A riffle occurs when water crashes over rocks, creating white water or bubbles. These bubbles introduce oxygen into the water, making this a perfect habitat for macros to thrive in. You can expect to find all different kinds of macros in this area, including stoneflies, mayflies, net-spinning caddisflies, and blackfly larvae.
So as you can see, sometimes riffles differ. Sometimes they're just like a rock down here sitting in the middle of the water. But if you look up there, we have full-size waterfalls. Like water's just pouring down. But it doesn't make a difference. Macros love to hang out in both. Sometimes you find the biggest stoneflies in the biggest waterfalls. So right below the waterfalls usually are deeper pools, and that's where Tyler is right now, just scraping off those rocks there and getting some of the possible big ones down in the depths. It's going to be a good scoop.
After sampling from a particular area, we dump our collection into the sieve bucket. A sieve bucket has a mesh netting at the bottom, allowing water to enter it when submerged in the water, but releases the water when you lift it out. This helps reduce the weight when walking through the streams. Also, the macros need water to stay alive, so it is vital to keep the sample submerged for optimum survival. Often, macros get stuck to the outside of our nets, so it's essential to visually inspect the net and make sure that they all made it into the sieve bucket.
Alright, so now we're basically going to invert the net into the seine bucket. But as you can see, there's still a lot of stuff attached to it. Lots of tiny macros like to hang on. And we are going to take a little piece of Tupperware, kind of wipe it off. Do it on the inside, do it on the outside. We're not gonna miss a spot on this thing. We make every sample count.
Other macros prefer different habitats to make their home, so we also check those habitats with varying techniques. We search through leaf packs that get snagged on the rocks, meaning they build up on top of one another as they fall from the trees, land in the stream, and flow down with the current. Commonly found macros in this habitat include cranefly larvae, scuds, caddisflies, and sobugs. We also check underneath snagged branches. A snag occurs when branches from the trees fall into the stream and get pinned between the rocks. These are excellent environments for macros because other organic material then gets stuck to the branch, creating a thriving microhabitat. We sample this area using the scrubbing technique shown here. Lastly, we search the undercut banks within the stream. Undercut banks are along the sides of a stream where the bank overhangs the water, as shown in this example here. Both dragonflies and damselflies tend to call this microhabitat home. After we make 20 collections from different habitats within the stream, it is time for us to start our sorting. First, we take the sample from the sieve and transfer it to a regular impervious bucket as we are doing here. We use small containers to wash out the sample, ensuring that each macro makes it out of the sieve bucket alive. This is now considered our primary sample.
Look at that. So much life hanging out on the sides. I can't wait to get in this soup of insects and invertebrates.
Once we do this, we make our way to the upper banks of the stream, take a seat, and then take subsamples from the primary sample and start sorting through to record what we find.
Like this, and we'll kind of stir it around. Mix it all up and then take out a sample. Take out some of the leaves, and because the sun is nice and bright, we can see really well if there's anything moving in here.
Each time we find a macro, we tally it on our sheet and use an ice cube tray to keep them separate from one another.
Collected. Some of the tools that we use are spoons and pipettes to collect anything we see moving. So you kind of just put the pipette in and kind of scoop up one of the little bugs. So if you look at your key, you can try to identify this. This is a midge. We collected quite a few midges in our sample here, so we'll put them with the other midges. Something else we see swimming around a lot in here are these tiny little ones. You see this guy? Let's see if we can get another one. It is quite small, but he has 3 pairs of legs. If you look really close, he has 3 tails. So I'll show you here in our field guide. These tiny little ones are actually mayflies. We actually collected quite a few. So let's move this to the side and see our ice cube tray where we kind of already sorted through them. So our mayflies are right in here. We have a lot of midges in this one. This one right here. All those kind of squirmy things, those are all midges. And they come in a few different colors and sizes. And this one, let's see if I'll scoop one up for you guys. This is a net spinning caddisfly. So in our key here, it's right down here. These are pollution sensitive. See that kind of C shape there? He's got 3 pairs of legs, some gills on his abdomen, and hardened plates by the head. And we actually collected 3 of these. So you may be wondering what this big one next to him is. This, kind of take a look at him, he's suction cupped onto the side of the ice cube tray there. This is actually a leech. Quite a large one too. These are pollution tolerant, so we find these in quite unhealthy streams. But since we have a diversity in this one where we had pollution intolerant, pollution sensitive, and pollution tolerant species, this is a great biodiversity that we have here. The one next to him here, this is another fun one. This one is a little bit easier to take out because he doesn't suction up. So this is actually a crane fly larva. So right here, pollution sensitive. These guys can get quite big, and they're also quite squishy too. Very fleshy body. So those are the macroinvertebrates that we found in our stream here today. And again,
We record every macroinvertebrate onto our data sheet until we reach a total of 100.
100.
This completes our biological assessment within our stream. Sometimes collecting and reaching 100 macros proves to be a challenge, and in that situation, we have to re-enter the stream and collect more until we reach our goal. It all depends upon the health within the stream, which is why we also perform a visual assessment of the habitats surrounding the stream. Many various sources could lead to a stressed stream, causing a poor habitat where not many macroinvertebrates live, especially up here in Hudson and Bergen counties. When you think of a pristine stream environment, they tend to exist in natural areas where not too much human intervention occurs. Examples include sediment deposition from construction sites that are building up condos to accommodate the growing population. This sediment introduces excess nutrients, changing the natural chemistry of the water. The sediment also covers the habitats beneath the surface, making it a challenge for sustainability. Deforestation, which removes the riparian forest that would naturally protect the stream. For example, think of a golf course and how open and green they are. To maintain their grass, the owners may spray pesticides to remove the insects and fertilizers to help the grass grow. Without that protective buffer of trees along the stream, anything they do to maintain the course will enter the water and negatively impact the quality downstream. Another example is channelization. This technique of humans deepening and modifying streams and rivers causes higher flows and increases the risk of downstream flooding. Many of these impacts are related. For example, straightening a stream channel can increase the stream velocities, the speed at which water flows, and destroy the downstream pool and riffle habitats, which is another reason why we may not reach our goal of collecting 100 macros. It is up to us to study the surrounding area, determine the biggest threat to the stream, and report our findings to the New Jersey Department of Environmental Protection. This is an example of a visual assessment performed on the Hackensack River near Bonnabel Nature Park. We use this sheet to score all of the varying stream characteristics on a scale of 20 to 1. 20 represents an optimal environment, whereas 1 would indicate a poor environment. Some examples of what we look for include epifaunal substrate. You want to see a healthy mix of cobble, snagged branches, leaf packs, undercut banks to provide cover for colonization of macroinvertebrates and fish. Sediment deposition, which is how much sediment has built up in the stream covering the habitat below the water. Bank stability, which is how eroded the stream banks are. And riparian vegetative zone. This is the area directly next to the stream. Ideally, you want a gap of 18 meters between the stream and human activity. Unfortunately, this is nearly impossible here in WMA5. Our last task is to take physical measurements of the stream we are assessing, and then our work is completed. First, we take measurements of the stream's width in 5 different areas and record those results in meters. After taking the measurements, we average that number out to get an idea of the overall width size of the stream. Next, we pick an area that represents the best overall combinations of depths and record 5 different measurements as we walk from the right bank to the left bank. Just like with the width measurements, we average this number and record the data. Lastly, we want to record the average speed within the stream. Using a rubber ducky, we stand 10 feet apart from one another and record how long it takes for the ducky to travel the distance. We repeat this 5 times and average that number to conclude the velocity within the stream.